สวัสดีครับอาจารย์ So today I am going to do the experiment about the ligating DNA fragment and also plating the transform bacteria. I am going to skip the transform bacteria experiment. The purpose of the experiment is to show or express the RFP gene in the E. coli bacteria.
Okay, so before we are going to do the experiment, I will show you the equipment used in this experiment first. So the first one is water bath. Second one is T20 micropipette. Third one is microcentrifuge. We also have the floating rack, and we also need the vortex. We need the P20 tips. We also need the freezer. So for the solutions, let's first start with the two solution tubes of digested pecan R and digested PRR. Both are stored in the ice bucket. We also need the DH2O distilled water tube, ligation buffer 5 times, DNA ligase tube, and the balancing tube. This is the rough procedure. The methods will be explained along with the video. Okay, so first thing first, we will move the K plus and the A plus tubes from the ice bucket to the floating tube rack. Then from the floating tube rack, we will move it into the water bath. Set the temperature of the water bath to be 80 degrees Celsius. Then we close the water bath and then set the time for about 20 minutes. Press start to begin the timer. After 20 minutes, remove the floating tube rack from the water bath and then we separate the K plus and the A plus tubes from the floating tube rack. Next, we will use the P20 micropipette. First, let's set the volume of the micropipette to be about 4 microliter. And then we will insert the tip into the P20. And we will start by draw up the A plus solution by placing the plunger down until it reaches the first stop. Then we will have to close the A plus solution tube, and then we have to open the DNA ligase tube and then dispense the A plus solution into the DNA ligase tube at the second stop. Then we will throw the usage tip of the P20 into the trash, and then we will follow the same micropipetting procedure to dispense 4 microliter of the K plus solution into the DNA ligase tube. Note that when we draw up the solution, we will hold it to the first stop, and then when we're dispensing it, we will hold it to the second stop. In order to make the DNA ligase to work at its best performance, ligation buffer are needed. Therefore, we will have to dispense 3 microliter of the ligation buffer from the 5X buffer tube into the DNA ligase tube. The addition of the 2 microliter of the distilled water are needed in the DNA ligase tube as well. First stop should be hold down before we put the tip into the solution, and we slowly release it when the tip is in the solution. Now we will use the vortex to mix the solution in the DNA ligase tube. After the DNA ligase tube is done in the vortex process, we will have to put it into the microcentrifuge. However, microcentrifuge also need the balance. Therefore, we will need to fill the balancing tube with the DH2O water. Therefore, the total volume of the DH2O in the balancing tube is 18 microliter, which is equal to the total volume of the DNA ligase tube. Balance tube should be put in the opposite way of the DNA ligase tube in the microcentrifuge in order to balancing it. Then we will push the pause button to spin the tubes. Once the spinning is complete, we will get the DNA ligase tube back into the tube rack. And then we'll put the DNA ligase tube at rest in the room temperature for about 10 minutes. Then we will have to store the A plus and the K plus tube in the floating rack, and then we will move it into the freezer for later analysis. This is the end of the 4.1 experiment, ligating the DNA fragment. Then we'll move to the next experiment. We should get this plasmid DNA if our experiment done successfully. Assuming that the transformed bacteria experiment is done perfectly well, now we are moving on to the plating transformed bacteria. The method will be explained along with the video. Reagents required in this experiment are P-positive and P-negative tubes. In the P-positive tube, it contains the E. coli competent cell with the intake of the PRR DNA from the previous experiment, and in the P-negative tube, it's just the competent E. coli cell without the intake of the DNA plasmid. Both of the tubes are mixed in the Rhodia broth. We will set the volume of the P200 micropipette to be 50 microliter. Then insert the tip for the P200 micropipette. Before we draw up any solution from the P-positive and P-negative tube, we have to mix the cells inside in both of the tubes first. Luckily, the P200 micropipette can do the mixing process. Draw up 50 microliter of the solution, and then we will dispense it into the P-negative side of the LB plate. Throw the used P200 tip into the biohazard bin. We use the cell spreader to spread the cells evenly across the entire P-negative side of the plate. Discard the spreader into the biohazard waste container every time we use it. Mix the P-positive solution. Draw up P-positive solution and dispense in the P-positive section on the LB plate. Use the cell spreader to spread the cell across entire P-positive section. Throw the cell spreader into the hazardous bin. Draw up P-negative solution and dispense into the LB-AMP plate on the P-negative side. Use the cell spreader to spread the cell across the P-negative side and the P-positive side. Always use the new cell spreader to spread the cell in every side and every plate. Do the same for the P-positive solution. Draw up the P-positive solution and dispense into the P-positive side on the LB-AMP plate.
to the bin.
Set the new volume of P200 micropipette into 100 microliter. Now we will draw up the solution from the P-positive tube, and then we will dispense it into the P-positive LB-AMP ARA plate. And then we will use the cell spreader to spread the cell across all the plate. Close all the lids, and then we will use a tape to seal the lids in all plates.
Like this. And
After all the lids are properly sealed, we will put them into the incubator, and then we will wait for 1 day or 24 hours, and then we will inspect the result. After 24 hours, we get all the plates back from the incubator, and then we inspect the result on each plate.
So in order to confirm if the experiment worked out or not, we will have to do the plating of the bacteria. So this one, if we take a good look, we will see a colony of white in the LB plate. This one we will not see the P negative colony, but we will see the white colony on P positive. And for this one, this is the purpose of this experiment. We want to see if the bacteria show the color red fluorescent as the colony, and this experiment show the red fluorescent color as the colony of bacteria for the E. coli. So it confirmed that this experiment worked out.
Thank you for your time and your attention.